Okay, in this video, we'll talk about the blood typing, and it's important to understand it because this is something we will use in blood transfusion. So we might have patients that they need blood because they lost blood, for example, in an accident or during labor, and we need to give them blood. So we need to make sure that the blood we are giving them would match their blood type. And to understand this, we need to do some drawing. So in the body, we have the cells of the body. Those are normal cells of the body. On their surface, they display proteins. Those proteins are used by the white blood cells to determine that those are cells or normal cells belong to the body. So those proteins on cell surface to identify cells for the white blood cells. So think, for example, the white blood cell would come and will check this protein. So it will see it that this is the normal protein or this is the correct protein. So it will not attack this cell because it will say this is like a self cell. But if this cell, if another cell is displaying a different type of protein, which the white blood cell would not recognize, then the white blood cell will understand that this is non-self or not self cell. And therefore this cell need to be killed. Okay, so those are the proteins. Therefore those proteins, we call them identifier or IDs. Okay, now a different name for them, a name that we need to be familiar with, is the antigen. So the antigen is a protein on cell surface to identify the cell. What do you mean by identify cell? To determine if it's a self cell or non-self cell. Okay, self cell, it means a cell that belongs to the body. Non-self cell, it means it's a cell that does not belong to the body. So this is in all over the cells, all of the cells of the body. Now for the red blood cells, those identifiers could be one of two. So those are four red blood cells, and those identifiers could be, we'll draw them like this, could be like this, a triangle, could be a square, could be a red blood cell displaying both the triangle and the square, or a red blood cell displays nothing, none. We'll just put the sticks. Okay, there is nothing on it. Let's call this triangle as A. So in this case, this red blood cell is displaying antigen A, and therefore this is A blood cell. This cell is displaying the square, we'll call this square B. So this cell is displaying antigen B. So this is a B cell. This cell is displaying both A and B. So it displays antigens A and B. Therefore, this is AB cell. And this cell displays none. Therefore, this is O cell. O means none, like think of the none. Okay? So those are the four types of the red blood cells in our body, depending on the antigen they present. Either they have antigen A, antigen B, both antigens, or none of the antigens. If it's antigen A, it's an A cell. If it's antigen B, it's a B cell. If both of them is AB. If none is O cell. So those antigens are critical in matching when we do the blood transfusion. In the old days, like how did we come to understand this? In the old days when they used to transfer blood between the people from one person to another person, sometimes this person will die and sometimes will survive. So it was like, why some people die, why some people survive, until they came up and discovered that there is mismatching of those white blood cells. Now let's try actually about the dying, why they will die. So we understand if I mix, if this person was blood A and I give it to a person that's blood A, this person will survive. But if this person was B, this person will die. So let's try to understand why they will die. Now let's go back to here. You see here when we said that the white blood cell would recognize this square, for example, as non-self cell. So it needs to kill this white blood cell, or it needs to kill this cell. One way to kill it is by secreting a group of proteins called antibodies. So this would secrete antibodies. And those antibodies like arms. Those antibodies will come, for example, let's assume that there is another cell displaying this non-self antigen, which was a square in our case. So those antibodies would come and bind this non-self antigen. Therefore, they will like kind of label this cell as non-self cell. So the monocytes or the macrophages will come and eat this cell. So the white blood cells would secrete those antibodies. So those will call them antibodies. And those antibodies, they are along with the red blood cells. So we have also, so in the blood, we have the red blood cells and we have the antibodies. So we have antibodies. And those are proteins for non-self antigens. So those are proteins for non-self antigens. What does this mean? Let's assume that we have a person. And this person has a red blood cell that is type A, which is, is displaying the triangle. In this case, this triangle is the self antigen. So does it make sense that I want to have an antibody for the self antigen? Or I need an antibody for the non-self antigen? What is the self antigen in this case? Let's assume that this is A blood type. So the self antigen is the triangle. And in this case, all of this person's red blood cells will have the triangle. All of them. None of them will display other antigen. No, all of them will display the same antigen because this is the self antigen. And in this case, the non-self antigen will be the square. So if I put a red blood cell that has the square, then this is non-self antigen or non-self cell in this case. Why? Because the self antigen is the triangle. Here the self antigen for this cell is the square. Therefore, this is non-self. It means it does not belong to this person. So I need the antibodies for the non-self antigens because those antibodies would come and bind the non-self antigen. Whereas the self antigen, those, if I have non-self antigen or like a self antibody that attacking them, we will be in big problem. So always the antibodies will be for the opposite blood type, for the blood type that we don't have, okay? Because I always looking for the non-self. I'm always looking for the non-self. I'm not looking for the self. Think, for example, in a company. In a company, each employee is carrying an ID, right? So all of them are carrying the same ID, right? This is the ID. So if there is a security guy, the security guy will be technically more focused toward a person that is carrying a different ID or displaying a different ID, not the square one like those, not the circle one. So this guy, the security guy, will look at this guy and try to take them out of the company. So this is how it works here exactly. Those antigens will work as this ID. And the body needs to look for the non-self, for the fake IDs or for the IDs they don't belong to this body. Therefore, we will have those antibodies. Antibodies against the other antigen. So let's go back to here. So if a person with antigen A, their body should have antibody that is against antigen B for this person. For this one, they should have an antibody for an antigen A. Therefore, we'll call this, in this case, anti-A antibody. And this one, we'll call it anti-B antibody. Okay, so always the antibody is the opposite of the antigen in the human being, inside the human being. Let's see if we can figure this out, the AB and the O. So let's assume that we have a human being, that their blood type is the AB. The AB. It means that they are displaying both antigens as self-antigen. So what are the antibodies in their system? Here in this case, both are self-antigens. So it means both are safe, both are belonging to the body, which means it doesn't make any sense to have any antibodies for them because if they are present, they will start to attack them, right? Which is something we don't want to happen because this will kill the person. So if a person is AB blood type, they don't have antibodies against A or B antigens. Okay? What about the O? If a person having an O red blood cell. O, it means there is no any antigen. Okay? So there is no self-antigen in this case. So it means that I need both antibodies because in this case, the square and the triangle both are non-self. So if I have a red blood cell that is displaying the square or the triangle, and the other one is displaying the square. Both are non-self because the self is displaying nothing, displays none. This is the self. So anything that's displaying anything else, we will consider non-self antigen. So those antigens will come and attack those cells and eliminate them. Okay? So here in this case, for this person, no antibodies for none of the antigens, but for this person would have both antibodies. Okay? So it's always important to remember that the antibodies in the human being, inside the human being, are always for the opposite antigen. Which means that if the person is B, then they should have an anti-A antibody, and so on. Now, we said for the blood type, we will have either A or B, or AB or O, none. But it turns out that there is a third antigen, and this third antigen will also affect the blood transfusion. So I will draw another red blood cell here, those red blood cells. In this case, we'll draw 6. Actually, we need 8. The third antigen, we'll call it the antigen D. And antigen D is coming, this was discovered in rhesus monkeys. That's why sometimes we'll call it like the RH antigen. And this RH, like there are 52 subclasses of them. The most important one is the antigen D. So in this case, we have, for the red blood cells, for the antigens, one of 3 possibilities. Either we will have the antigen A, or the antigen B, or the antigen D. And those could be on any of the red blood cells. So for example, we can have A with D. So in this case, we'll call it A because A is here. And for the D, it's either present or is not present. So if it's present, we will call it A plus. So the plus sign, it means that antigen D is present. If it wasn't present, like here for example, we'll call it A minus. Okay, A minus. It means that antigen D is not there. Usually for the plus, 85% of the Americans, they have the antigen D, 15% they don't have the antigen D. And therefore for the blood group, technically we have 8 blood groups depending on the presence of the D. So here in this example, we have the A plus, A minus, we have the B plus and the B minus. So this is B, this is B. So either we have it or we don't have it. In this case, this is the B plus, this is the B minus. We have the O, either we have it on the O or we don't have it. So in this case, this will be the O plus and this is the O minus. For the AB, either we have it or we don't have it, but with the AB, we have the other 2, right? We have the other 2. So in this case, it is AB plus because of the D. Here it's AB minus because it's absent. Okay, so those are the 8 blood groups that we usually need to do transfusion across them. So for the blood transfusion, it's easy as long as we remember the combination. So usually for the blood groups, each blood type can give its own. So for example, we have the A can give itself, okay, and it can give the AB. B can give also the AB and can give itself, okay. The O can give to A, can give to AB, can give to B. Why? The O doesn't display any antigen. So if I go to the people here, to the person here, okay, I will erase all of this. Or actually, let's draw a different person. So this person is O. Let's assume that it is O minus. Okay, so this is O minus. So it means that there is no A, no B, no D on the surface. So in this case, this one, okay, if I have another person, let's say that this person was A plus. And if I put this red blood cell in there, it doesn't display any antigen. So the antibodies will not be able to recognize it. Okay, so therefore the O can give to all of the blood cells, to all of the blood type. And of course, the O can give to itself. Always the letter can give to itself. But however, for the AB, it can only give to itself. The AB cannot give to the A, cannot give to the B, cannot definitely give to the O. Nobody can give the O except itself. Therefore, the O is the universal donor, whereas the AB is the universal recipient. For the plus minus, the plus can take from itself, okay? The minus can take from itself, and the minus can give to the positive. The negative can give to the positive. It means if, for example, A negative, it can give to A positive, but the A positive cannot give to the A negative. I need you to think why. Okay, why the positive cannot give to the negative, and we might talk about it, or actually we will talk about it in the lecture. The same thing, why AB plus cannot give AB minus. Why the positive cannot give the negative. Okay, but this is for the blood typing. Now, for the blood group detection, this is something we will do it in the lab. And what we will do, we will have 3 wells. In those wells, we will have the antibodies. So here in this well, we will have the antibody A, which is like this. We will have the antibody B, and we will have the antibody D. And we will throw the red blood cells. And we will see which antibody would catch something in the red blood cells. So if there is catching, it means there is agglutination. What's happening is that the red blood or the antibody can catch several of those cells at the same time. So that way we will have the agglutination, and therefore we will know that those red blood cells contain the A. So if we have the agglutination, it will take its letter. So here we have agglutination, we have clotting in the anti-A. So this will be A group. No agglutination in the B, so it's not a B group, but there is an agglutination in the D group, so this will be positive. Therefore, this will be an A positive blood type. Okay, so always remember, if there is an agglutination, the blood type will take the letter. So there is agglutination in A, it will take the letter. No agglutination in B, it will not take the letter. Agglutination in D, so instead of saying D, we'll say positive. Okay, and this is something we will talk about it in the lab. We will do the blood typing in the lab, and there we will understand what exactly we are doing, because here, to understand it, you need to reverse what we just learned. Okay, we need to reverse what we just learned. One thing to think about it until we are in the lab, think, for example, if there is a person wants to buy shoes, and this person doesn't know their shoes size, so they will go to the shop and test, for example, size 10, they will test size 11, and they will test size 12. So if their feet is not perfect in size 10, then their size is not 10. If their feet is perfect on the 11, then their size is 11. If their feet is perfect in 12, so maybe also they are, size 12. Okay? Or maybe it's not. So it will be just size 11 in this case. So that's exactly what we are doing here, because we don't see the antibodies on the surface of the cell. So we will test the cells against the antibodies. We will see which antibody will catch. And if the antibody catches something, it means that thing is there, or the opposite antigen is there. It's exactly like here. I have the template, which is the shoes. The shoes is size 10. Okay? If the feet is size 10, the feet will fit into the shoes. If they are not, then the feet are not size 10. So it's kind of reverse engineering. I will know the size of the feet by the size of the shoes. Okay? So that's the blood group. The last thing in this chapter is something called hemolytic disease of the newborn. And here, this is related to the Rh antigen, or to the positive minus antigens. So this is not about the A or B. No, this is about the plus or minus, regardless if it's A or B or AB or O. So what's happening, let's go back to do some drawing as well. So let's assume we have a pregnant woman. This woman is having O minus. Okay, her blood group is O minus. But she's pregnant with a baby, and the baby, let's assume, for simplicity, we'll assume that it is O positive. O positive. To understand what's happening, what are the antibodies in the mom in this case? We will have antibodies against A, against B, and against D. Okay? So the 3 types of antibodies, because she is not displaying any antigen. So always the antibodies are for the missing antigens. Okay? So during pregnancy, some of those red blood cells of the baby would escape through the placenta to the mom. So we will have suddenly non-self cells in the mom system. And the antibodies will start to attack those red blood cells and eliminate them. So sometimes we'll get maybe like 20, 30 of those red blood cells. So, and the body will deal with them as an infection. So this is an infection. So we'll eliminate them. So in the first pregnancy, it's no problem. The body will eliminate those red blood cells. The problem would be on the second pregnancy. So again, let's assume that the mom is pregnant, and again, the mom is still O minus. In her system, we still have the 3 types of antibodies. And now the baby is again O positive. And now some of those red blood cells would escape to the placenta. Now the body will not only this time deal with those red blood cells, but will start to send those antibodies into the infant, into the fetus. Because the body will consider the entire fetus, in this case, is an infection, and it needs to eliminate them. Okay, so it will start to attack the pregnancy itself, and this of course will cause the baby to die even before birth, which will cause complications for the mom as well. Okay, so that is the new hemolytic disease, or the hemolytic disease for the newborn babies. And usually this will happen on the second pregnancy of negative moms, or moms with negative blood type, with positive blood type babies. Only in this setting that the mom has negative blood type and the baby has a positive blood type. That is when we have the hemolytic newborn disease, or hemolytic disease of the newborn, HDN. Okay, so now what they are giving, they will give like an injection, like a vaccine that would tell the body to tolerate some of those red blood cells. So here, for example, this is the placenta. This is on the first pregnancy. Those red blood cells escape, and the body will deal with them, will secrete those antibodies. But again, on the second pregnancy, those antibodies will start to attack the fetal circulation, okay, the infant blood, and that's what will kill the infant. So usually now they will give the mom, especially the mom with negative blood groups, they will give them a vaccine. This is called RHO-GAM. Okay, this for Rh immunoglobulin, which will temporarily prevent the development of Rh antibodies in case of Rh positive. Okay, so this is the end of this chapter. Let me know if you have any questions.